So whenever we are talking about determining the level of antimicrobial agent's activity, we need to apply certain tests that can show how effective a particular type of antimicrobial agent or an antibiotic is against the colony of bacteria that we are testing. So in today's lecture, which is this in the first section, we'll be discussing the antimicrobial agent's effectiveness. There are a number of tests that can help us identify if a bacteria is more resistant or more susceptible to a type of antimicrobial agent that is usually used to stop its growth or to limit its growth. We by now know the concept of the minimal inhibitory concentration and the minimum bacteriostatic concentration. Both of these levels are actually the crucial points on which we can, on which the tests are usually based, because whether you're using broth or agar to grow your bacteria, at the end of the day, what you're more concerned about is the amount of antimicrobial agent that is required to stop the growth or to inhibit the growth. And obviously we're talking about the minimum amount, because sometimes if you're using more amount of antimicrobial agent to stop the growth, you might stop the growth, but you can damage the surface, or you can damage the person who's taking the antimicrobial agents or the antibiotics to treat a particular type of bacteria that's growing inside him. So we need to identify the safe zone in which the antimicrobial agent can be effective enough to stop the growth of the bacteria, but not toxic for the surface or for the person or the host who's taking the antimicrobial agent. So in this particular section, we'll talk about the antimicrobial agent effectiveness by using a test which is known as dilution susceptibility test. As the name suggests, this test basically helps us identify the minimum concentration. We keep on diluting until we find a level beyond which, or after diluting, which the antimicrobial agent loses its ability to stop the growth. There are two further tests in a dilution susceptibility test, that are the broth dilution test and the agar dilution test. There are certain bacteria that grows better on the liquid medium as opposed to the solid agar, while others required a solid substrate to grow properly on. So the dilution tests or the principle behind the dilution test is similar, but the broth and agar depends on the type of bacteria or the colony of bacteria that you need to grow. So starting with the determination of antimicrobial agent effectiveness, this is essential for proper therapy, as I've suggested earlier. Whenever you're talking about an antimicrobial agent, you're talking about killing a type of bacterial growth. Especially, we are more concerned about the bacteria that grows inside a person's or an organism's body. So whenever you're talking about using an antimicrobial agent to stop the growth in the host, you need to determine the minimum amount that is required, that can stop the growth but cannot be toxic to the host. And so it's essential for the proper therapy of a person to know the exact amount that is ideal, and that can actually stop the growth of bacteria without damaging the cells. So the testing that we usually apply to check the antimicrobial agent effectiveness can show which agents are most effective against a pathogen. As I've said earlier, there are certain pathogens that can be readily killed or their growth can be readily stopped by using an antimicrobial agent, but there are others that can be resistant to it and can resist even a high dosage of that particular antimicrobial agent. So the testing before using or before sending an antimicrobial agent to the market is important and crucial, because you need to determine its lethal dose, you need to determine its MIC, its MBC, and you need to determine the set of pathogens that it can actually work against. So this gives a proper estimate of therapeutic dose, which is the ideal dose that can kill the microbe without damaging the host. So whenever we are talking about selecting a particular type of antibiotic, the first thing that we need to understand is the type of pathogen that you need to that you're targeting. After knowing the pathogen, so that's why usually whenever you go to the doctor or see a doctor, the first thing that he usually do is get a test of or get a sample for testing before prescribing the antibiotics usually, or usually give you a wider range of antibiotics, so that if there are a number of pathogens that are present that can cause a particular problem, then the antibiotic should cover all the range of the pathogens. So whenever you're determining the type of antibiotic you need to use, initially you need to know the pathogen of interest. The antibiotic selection is then dependent on the pathogen. Then there is a fractional factorial design, which means that you need to understand and identify if a particular antimicrobial agent itself is sufficient enough, or if you need to make a cocktail of antimicrobial agent which can act together, have a synergistic activity, and can kill the pathogen. And usually cocktails are given to increase the range of pathogens that need to be killed, because like I said earlier, we are not usually talking about a very pure colony in which only one type of pathogen is growing. Rather, there are usually a number of species in a particular surface, so we need to have a wider ranged antibiotic. So in dilution susceptibility test, the MIC and MLC values are used, or the testing is used to determine the MIC and MLC value. The antibiotic dilution test can be done in both agar and broth. So this depends on the type of bacteria that is to be grown. It can be in broth or in agar. As the name suggests, we are talking about the dilution, which means that we are talking about diluting the amount of antimicrobial agent in broth or in agar, and then we see the minimum amount in which there is no growth can be seen. So as you can see, when you're decreasing the antimicrobial agent beyond that amount, when you're decreasing it, there is growth that can be easily seen in the broth. But when you increase it, there is no growth. So the minimum amount, as the figure shows in this case, is 2 micrograms per ml. This is the amount in which no bacteria is growing, and beyond this, that is when you lower the concentration of antimicrobial agent further to 1 microgram per ml, you can see that growth can be seen. So the MIC is the center point. Decreasing the level of antimicrobial agent beyond this point will cause growth. So then usually what we do is we inoculate from the MIC cultures into antimicrobial free agents to see if there is bacterial growth, and usually there is bacterial growth because in MIC there is a minimum inhibitory concentration, which means that the antimicrobial agent is not entirely killing the pathogens that are present, but rather stopping their growth. So to determine the minimum bacteriostatic concentration, what we usually do is we take a sample or an inoculum from the MIC, and then we inoculate it in a broth that is free of any antimicrobial agent, just to see that there is whether there is a growth or not, and beyond which point the growth is stopped. So In broad dilution test, we are again using MIC to determine the effectiveness or the activity of an antimicrobial agent. The lowest concentration of antibiotic, which result in no growth after 16 to 20 hours of incubation, is the minimum inhibitory concentration. So whenever we are talking about an MIC, it means that we have given sufficient time, that is 16 to 20 hours, to the antibiotic to stop the growth of antimicrobial agent or the microbes, because it's not like you can apply the antibiotic and expect result or expect complete killing of bacteria right away. So there are a series of tubes that are used. The broth which is used in these tubes is called Muller-Hinton broth. So this broth has all the required salts and all the required minerals that can be needed by the bacteria to grow ideally. So this is an ideal broth that can readily help bacteria grow. Then the antibiotics are added to the broth in a range of 0.1 to 128. So this basically means that the antibiotic range varies greatly, and even a point difference of antibiotic can either stop the growth or can let the bacteria grow. So it's very important to make the dilutions in such a way that you don't miss out the point, which is the MIC. So even a point of increase or decrease of antimicrobial agent matters in this case. And then they are inoculated with standard density of the test organism that needs to be checked. If you look at the figure, you can see that you take the cultured organism, then you grow them in broth, then you add them to the tubes. Tubes with increasing drugs concentrations are inoculated with the standard number of organisms. So it is important to know that the number of organisms that you're taking is going to be the same for each tube, no matter the concentration of antimicrobial agents. And then you need to determine the MIC, which is the minimum drug concentration, which will inhibit the bacterial growth. And when there is no visible growth, as you can see in this case, MIC is 8 microgram per ml. And you just pick up colonies from there and you grow them again. And when you see a zone where there is no growth when plated, then that is the MBC, which is the minimum drug concentration, which actually kills the entire bacteria. So what we are doing is we are taking the colonies from all the tubes, and then we are plating them again. And then the colony or the sample which shows no growth is the amount of MBC, which in this case is 16 micrograms per ml. So this just shows what MLC is. If tubes showing no growth are cultured into fresh medium that lacks antibiotic, then the lowest antibiotic concentration from which microbes do not subsequently grow is the MLC. Again, the figure shows how you can check for the MIC and MLC, and how you can also use controls in which there can be no antibiotics to check the viability of bacteria on agar plates. And in control 2, you can add no bacteria to see that there's no other pathogen that's growing beside your own pathogen of interest. So you can see that the bacterial once the bacterial colonies are inoculated, they are incubated, usually at 35 to 37 degrees. That's the ideal condition for bacteria to grow. And once they're inoculated, they are incubated for usually overnight, and then we can determine the MIC and MLC. Then the other test is the agar dilution test. It's pretty similar to the broad test because we are using we are diluting again in this situation, but we are using plates. Plates contain the Muller-Hinton agar. So even the concentration of the vitamins or minerals that are present in the broth remains the same, but it's in the solidified form. And some amounts of antibiotics are inoculated and then examined for growth. So you can see that there can be plates with different amount of antibiotics, and some of them will show various bacterial colonies growing on them. And obviously you can have control in this situation as well, in which you can use a plate with no antibiotic as a standard, and you can use plate with no inoculation of bacteria as a standard. And then obviously there are other plates in which there are different concentrations of antimicrobial agents, and the plate where there is no growth that can be seen, that usually help us identify the antimicrobial agent's activity. So there are automated systems that are used for susceptibility testing and MIC determination in broth and agar. So with the growth of science and with the growing technology, now there are automated systems that can read the MIC and MLC for you, and you just have to insert the broth or the plates or the tubes in the system, and the system will tell you the minimum inhibition concentration of a particular antimicrobial agent, and you can then take samples from there to determine the MBC as well. So in this particular section, we talked about one of the tests that is required or that is commonly used to check the effectiveness of antimicrobial agents. That's all from this section. Thank you for watching Scadia.com.